Bom dia, pessoal. Nós iniciamos novamente nosso curso, hoje contando com a presença de John Archibald. É um prazer recebê-lo aqui hoje, ele está aqui já conosco. E, primeiramente, eu vou fazer uma apresentação curta do currículo do professor John. O professor John ele é diretor do Center for Comparative Genomics and Evolutionary Bioinformatics da Dalhousie University desde 2017. E ele é professor da Dalhousie University, ministrando aulas de bioquímica e biologia molecular. Ele é membro do corpo editorial de uma série de revistas, como Current Biology, BMC Biology, Environmental Microbiology, e ele é editor associado também da Genome Biology and Evolution. Ele conta com mais de 156 publicações científicas e mais de 95 palestras, coordenador de uma série de projetos e portador de uma série de condecorações por diversos prêmios. Uma delas aí, como membro eleito da Royal Society of Canada. Ele já supervisionou 21 projetos de pós-doutorado, 11 pesquisadores associados e assistentes, 11 doutorandos e 21 estudantes de graduação, que seria equivalente à nossa graduação, além de 3 estudantes do ensino médio, aquilo que é equivalente ao nosso ensino médio. Então, com vocês hoje, tratando sobre a endossimbiose e a origem dos plastídeos, professor John Archibald. Thanks for coming and present us with your presentation and your experience in this theme. Thank you so much.
Well, thank you very much, Professor Silva. It's really great to be able to connect with you and with such a broad audience. In particular, it's always a pleasure to interact with students at various levels, from undergraduate, graduate students, and so forth. It's a part of my job that I really enjoy, so I hope that you will get something out of my 2 talks. In terms of the structure, what I'm going to talk about today, it's split into two different parts. And the first one will be about 50 minutes or so, with time for questions. As I mentioned to Wellington, I'm perfectly happy for people to ask questions as I go along. So if I'm speaking perhaps a bit too quickly and you'd like clarification on something, you're more than welcome to interrupt and ask a question about a particular point. I'm more than happy to do that, and we can adjust the timing of the lectures accordingly. Okay.
Okay.
So if you could go to the next slide, please. My longstanding interest in research really has revolved around eukaryotic cells and their organelles, in particular chloroplasts or plastids. And this picture is one of my favorite in all of science. In the bottom left, that's a cross section through a plant leaf, and of course those little green balls are chloroplasts. And we know that those subcellular entities evolved from once free-living cyanobacteria. This has been, it's now established scientific fact, and it's very difficult to overstate the impact of that endosymbiotic event on life on this planet. In the top right is an image of my part of the world. You can see land masses there are green, of course, and so photosynthetic life colonized Earth and the land, and this contributed to the greening of our planet Earth, and of course many of the organisms that I'm going to talk about today, and that that you're interested in as well. The algae are marine organisms, and what you can see in the bottom right of that upper right picture is a haptophyte algal bloom off the coast of Newfoundland. So there's really no doubt that photosynthetic eukaryotes have had a massive impact on the trajectory of planet Earth. That red arrow, incidentally, is pointing to Halifax, Nova Scotia, which is where I'm sitting right now. So we'll go to the next slide, please. What I'm going to do for the first part of this first lecture is tell a little bit of history. And the reason I like to do that is because it's very easy for people who have been educated in the molecular and genomic era to simply assume that what we know today has been known forever. And in the case of organelles, the idea that, well, we know that chloroplasts in mitochondria are of endosymbiotic origin. We know this because of lots of different reasons, including the fact that their genomes tell us that this is the case. But in fact, that's a relatively recent realization and discovery in the history of science. And so what I'm going to do is tell you a little bit of the backstory, the sort of pre-molecular history of how it is that people have come to realize that mitochondria and chloroplasts are of endosymbiotic origin. And then the second part of this first lecture is going to cover some modern perspectives on what we know and still don't know about the evolution of plastids from cyanobacteria. Okay, next slide. So many of you, I suspect, will have heard of Lynn Margulis, a famous American scientist who passed away in 2011. She is, in many circles, synonymous with the concept of symbiosis in biology. I'll just read this quote because I think it accurately captures her sort of stature and prominence in biology. Like the eventual acceptance of continental drift, acceptance of a symbiotic theory of cell evolution has often been hailed as a scientific revolution. The woman most responsible for bringing the idea to scientific respectability is Lynn Margulis. A prolific writer and dynamic speaker, Margulis captivates audiences and often irritates more traditional biologists with her unorthodox ideas. Although many biologists continue to disagree with some of her ideas, everyone takes endosymbiosis seriously. And it is indeed because of her massive effort to engage with the public, to emphasize the importance of symbiosis in biology, that even non-specialists in different fields of science have probably heard of Lynn Margulis. Next slide, please. What many people will be less familiar with, and even within biology, practicing biologists, that this idea of symbiosis is very, very old, and in fact was around long before Lynn Margulis championed the idea. The definition of symbiosis in biology, there are lots of different variations on that theme, but one simple one that's quite effective is the living together of unlike organisms. And it was a Swiss biologist, Simon Schwendener, who in the mid-1800s or so first started to articulate ideas of symbiosis and to bring them to the attention of other scientists. And he put forward this idea of the dual hypothesis of lichens. So many of you will know these lichens, and it's a picture of them there. Rather unassuming organisms in many cases, although those ones are quite colorful. They are in fact not a single organism. So a lichen is not one organism. It is a symbiotic relationship between two organisms, a fungus on one hand and an alga on the other. And so this was quite a controversial idea at the time, because the idea that an organism, a lichen, say, could be actually comprised of two or more organisms was somewhat heretical and not particularly well received. In the case of lichens, you may know that they have evolved many times during the course of evolution, and in some cases you can tease apart the two partner organisms, the alga and the fungus, grow them separately, and then stick them back together to reform the lichen. And so this was hugely problematic for people trying to understand and to categorize the living world. How can a single thing be composed of multiple organisms? Incidentally, we know today that lichens are, in fact, many lichens are composed of three partner organisms, two different flavors of fungus, and either a cyanobacterium or a green alga. So it's more complicated even than Schwendener had envisioned. Next slide, please. Now, there's a rich history of symbiosis research in Russia in the late 1800s into the early 1900s, and these three individuals here are often referred to as symbiogeneticists. I'm going to talk only about one of them briefly, the individual in the middle, Konstantin Mereshkovsky. Next slide, please. Now, Mereshkovsky published in hindsight, remarkable paper in September 1905, and it is there in German. We'll go to the next slide. Fortunately, that paper was translated quite recently into English by Martin and Kowalik. The title of that 1905 paper essentially is on the nature and origin of chromatophores, chloroplasts, that's the word for them at that time, in the plant kingdom. And it's interesting to just think a little bit about some of the evidence that Mereshkovsky brought forward to make a case for the fact that these green entities within plant and algal cells were related to cyanobacteria. Now, Mereshkovsky was broadly read and studied lots of different organisms. He was a specialist on diatoms, as it turns out. So he was aware of symbiosis as a general phenomenon that people had postulated. And he was aware, too, of lichens and their complex biology. He also knew that people such as Andreas Schimper in the late 1880s had documented the fact that chloroplasts, or plastids, I'll use those words interchangeably, those subcellular entities proliferate by division. So daughter chloroplasts come from mother chloroplasts, as it were. And he referred to that as the continuity of chromatophores. So chloroplasts are not created from scratch by a eukaryotic cell. They come from pre-existing chloroplasts. So those entities divide within the context of a plant or algal cell. He was aware of much of the high-level physiology of chloroplasts, so the way that they carry out photosynthesis, and he noted similarities between chloroplasts and cyanobacteria. So oxygenic photosynthesis happening in both of those two biological entities, and that was a connection that was meaningful to him and people who came after him. And so he postulated that, in fact, these cyanophyceae, so cyanobacteria, are nothing more than free-living versions of chromatophores, chloroplasts. And again, looping back to his broad knowledge of biology, he knew that there were other types of examples beyond well-studied plants and algae that had cyanobacteria living inside of them. A particular organism called Paulinella was of interest to him, and I'm going to loop back to that topic in a later slide. So we'll go to the next slide, please. Now, in hindsight, it's easy to consider Merezhkovsky's 1905 paper as a real landmark in biology, and many people in the field would say that today. But at the time it was published, it was largely ignored. So there were different camps of researchers in different places working on this problem and question. One of those individuals was a Frenchman named Paul Portier, and in 1918 he wrote a book called Les Symbiotes. And this was a book that revolved around important properties of mitochondria. And to make a long story short, he believed that mitochondria are not just derived from bacteria, but mitochondria actually are bacteria. And complex life forms such as ourselves, multicellular organisms, actually depend on these intracellular bacteria for life. And he linked all manner of important biological processes to these mitochondria, everything from development to speciation, and even to cancer, things like cancer as being disruptions in the biology of mitochondria. Next slide, please. Now, unfortunately, Portier's paper was published in 1918, and a year later, a review of that, a sort of rebuttal to that came out, was published by someone named Auguste Lumière, and he was one of the people, he and his brother invented the motion picture camera of all things. Lumière is famous for other reasons than this book, but his book was entitled Le Mythe des Symbiotes, and you can guess from the title that it was not an endorsement of Portier's Les Symbiotes. It was a critical critique, and long story short, Portier's idea that mitochondria were important, and in fact bacteria living together with eukaryotic cells was largely dismissed. This is a quote I won't read you, but an anonymous review in Nature, which basically said that we have no doubt that Portier's ideas will be proven false in the near future. Next slide. Now, on the other side of the Atlantic Ocean was an American anatomist named Ivan Wallin. And Wallin taught medical students for a living, turns out. But on the side, he was infatuated with mitochondria, and he wrote a series of papers and published a book, and that's the cover of it there in 1927, Symbionticism and the Origin of Species. And his role in symbiotic theory is somewhat controversial and contradictory depending on the sources, but he was very critical of the work that Portier had done, but also propagated some false ideas forward in time, unfortunately. Next slide. But one of the things that Wallin insisted upon, as Portier did, was that mitochondria were, again, they're not derived from bacteria, they are bacteria. And what Wallin did was to carry out comprehensive experiments to show, he thought, that mitochondria could be cultured. So he figured this was the ultimate proof for the connection between mitochondria and bacteria. So he would take animal tissues, grind them up, and plate out the extracts on agar plates and observe bacterial colonies. And that's a picture from one of his original notebooks. And so you can see the pictures of the colonies. In the fullness of time, of course, we know that this is not true. It is impossible to culture mitochondria because they are fully integrated within the confines of the eukaryotic cell. So what Ivan Wallen was no doubt culturing were simply bacterial contaminants that were floating around in the air or on his plates or what have you. And really his legacy is a lengthy set of papers and a book that really were just hinting at the right direction, but for all the wrong reasons. So Wallen is a much maligned and discussed figure in the annals of symbiosis research. Next slide. So the idea that mitochondria and chloroplasts could be of bacterial origin, as I hope I've convinced you, it's been around for a long time, but it took an awful long time for people to actually start to take it seriously. And there are various reasons for that. One reason was the fact that if you think about the time where the history of science and biology was heading into towards the mid-1950s, people were still trying to figure out the nature of heredity. And geneticists like Morgan knew that chromosomes were in the nucleus, even though we didn't necessarily know that DNA was the genetic material. We knew that chromosomes, which are of course DNA and protein, were the seat of heredity. So Morgan knew and told people in his work that the nucleus is where it's at in terms of heredity, and basically the cytoplasm can be ignored genetically. And of course, the cytoplasm is precisely where mitochondria and chloroplasts reside. And so if you didn't really have any vested interest in this question, it was enough to simply take Morgan at his word. There are other scientists in that general era who were quite familiar with the work of Merezhkovsky and Wallen. One of them was a cell biologist named Wilson, who is the publisher of what's thought to be the first cell biology textbook. And as you can see from those quotes, he was rather dismissive of these ideas that organelles could be derived from free-living bacteria. So he was aware of the earlier work of Merezhkovsky and Wallen. He just didn't find it particularly compelling. Next slide. So back to Margulis. Well, she came of age scientifically at a very critical time in the history of biology and in particular molecular biology. So she worked with important people who were experimentalists. So her graduate, her master's and PhD degrees were experimental in nature, even though she's not known from that point on for her experimental work. She's much more known as a theoretician and a popularizer. She did work on algae such as Euglena and on various amoebae, and provided some indirect evidence for the existence of nucleic acids in the cytoplasm, which again pointing to the idea that mitochondria and chloroplasts have DNA within them. Next slide. So really the technologies that were coming of age at that time, electron microscopy. So some of her PhD advisors were the very first people to perform detailed sections by TEM and to demonstrate that organelles actually contain DNA. So these were some of her mentors in her graduate days, and so she started to connect the dots, so to speak, and to put together the pieces to make a compelling case for the endosymbiotic origin of mitochondria and chloroplasts. She published her first paper on this particular topic in 1967. At that time, her last name was Sagan, or Sagan, as you wish, Lynn Sagan. She was married for a short period of time to Carl Sagan, the astronomer and science popularizer. And she posited that not two, but in fact 3 different subcellular organelles were of endosymbiotic origin. So the 2 we've already talked about, mitochondria and chloroplasts, but also the flagella, the flagellar apparatus of eukaryotes. She posited that that was derived from spirochete bacteria. Next slide. In 1970, she published the book that really made her famous, and that's The Origin of Eukaryotic Cells. That's the cover of it there. And this was a real attempt to reach out to the broader scientific community to tell them all about the importance of symbiosis in eukaryotic evolution. Next slide. Now, to contextualize this a little bit and give you a sense for why this is so important, the different perspectives on where organelles come from, you have to think about what the more common views were at the time. So if people didn't think that organelles were of endosymbiotic origin, what was it that they did think where they came from? Well, the tree on the left, it's just a cartoon, of course, but it illustrates the diversification of eukaryotes from prokaryotic predecessors. And in particular, the prevailing view was that all eukaryotic life came from blue-green algae, from the cyanobacteria. Okay, and the idea here is that the ancestor of all eukaryotes was a photosynthetic organism, and that secondary loss of chloroplasts and photosynthesis had given rise to animals and fungi and all of the other organisms that do not have chloroplasts. Now, Margulis didn't mince words. She referred to this as the botanical myth because it was the botanists who were the ones positing this vertical scenario for the evolution of eukaryotes. And on the right is her cartoon showing endosymbiotic origins of 3 parts of the cell: mitochondria, chloroplasts, and the flagellum. Okay, next slide. Now, this is where the molecular sequencing technology started to come in. And I'm going to tell you very briefly about how this came to pass. Now, the individual on the left, you might recognize, that's Carl Woese, the discoverer of the archaea or archaebacteria. And he had been developing technologies for sequencing small RNA molecules, namely to try and understand novel lineages within the prokaryotic domain. And long story short, he and someone named Linda Bonin, who's now a retired professor here in Canada, applied this technology to look at organelles. And RNA cataloging is a rather complicated, very laborious technique. At the time, there was only a handful of people on the planet, really, who could carry out the experiments and actually interpret the data. It involved isolating huge amounts of RNA, radio-labeled RNA, and separating it on 2D gels, and then being able to digest the spots, isolate the spots, digest them with additional enzymes, and look at their migration patterns, and so on, and ultimately be able to produce these little snippets of RNA sequences. You can see there are 6 or 8 nucleotides long, and from that you could start to produce, to carry out sequence comparisons to try and understand how bacteria related to one another and how organelles were related to bacteria. So next slide. Now, Bonin took that technology to the lab of Ford Doolittle and Michael Gray, and they happened to be working here in Halifax. They still are. At that time in the early 70s, they were doing pioneering work on lots of different things, but one of the things they did was to obtain some of the very first RNA and then DNA sequence data from mitochondria on the one hand and chloroplasts, and the results were very strongly pointing towards the fact that these organelles are very clearly the residue of DNA in the chloroplasts and mitochondria had strong affinities to the to free-living organisms, proteobacteria on the one hand and cyanobacteria on the other. Okay, next slide, please. This is just a montage of some titles that came out of a symposium in 1980. Now, it would be tempting at this point, looking back, to think by 1980, when there were lots of molecular sequence data around, that the case would have been solved. That is, everyone would agree with the molecular data, but in fact, that was not the case. There was a long period of time where there were two different camps, those who believed that chloroplasts and mitochondria were endosymbiotically derived, and those that did not. And even with molecular data in hand, many people took to producing alternative interpretation of those data. So it wasn't a very extremely rapid switch in terms of the field changing their mind. It took sort of the better part of a decade for the idea to take hold. So next slide. And this is a 1982 paper, which is a very, very highly cited review article, and it covers all of the evidence that was put forward in the early 80s that supported the idea that chloroplasts and mitochondria are endosymbiotic in origin. It's phylogenetics, biochemistry, molecular biology, and organelle ultrastructure. So next slide, please. Now, to summarize all of this, I hope I've convinced you that it's actually very difficult to have a truly original idea in science and of course, in life. In the case of Lynn Margulis, timing was everything. So she was coming of age as a scientist at a time when new tools and technologies were able to be applied to this problem. And she took it upon herself to synthesize very diverse lines of evidence to create a picture pointing to the idea that the endosymbiotic theory really does account for organelle origins. But the last point is to say that it is possible to have too much of a good thing. Next slide. And by that I mean that she proposed that there were in fact 3 different organelles of endosymbiotic origin. Wellerton, if you could go to the next slide, please. Yeah. So including the flagellum. So she, that was one of her original formulation of symbiotic theory, had that eukaryotic flagella were of endosymbiotic origin, and that has never taken hold as an idea. And she actually believed that throughout her career, but had very little in the way of evidence to support it. So endosymbiosis explains a lot about the membrane-bound compartments in eukaryotes, but it doesn't explain everything. Pretty much every compartment in a eukaryote has at one time or another over 100 years of science has been proposed to be of endosymbiotic origin, but really only the chloroplasts and mitochondria have stood the test of time. And this is largely due to the fact that they have nucleic acids inside of them that we can study in terms of their molecular sequences. Next slide. So let's move on to the last point, which is to dive into the sort of current perspectives of where our understandings of the evolution of plastids or chloroplasts come from. Next slide. And this audience needs no introduction to the incredible diversity of photosynthetic eukaryotes on Earth. And so what I'm going to do is start to chronicle through some of these groups from an evolutionary perspective, beginning with what's referred to as the primary endosymbiotic origin of chloroplasts. So next slide. From the beginning, we can paint a very clear picture of a broad picture, but one that has stood the test of time, which is that cyanobacteria were the prokaryotic progenitors of chloroplasts, and that the host organism was a fully fledged eukaryote of some kind. And you will also know that there are three main lines of extant photosynthetic life that have plastids derived from this primary endosymbiotic event. In the middle is the green line. So these are the green algae and their land plant derivatives. I gather you're going to hear a talk from Charles Delwich later on in this course talking about land plant evolution. So that's the green line. There are, of course, red algae, a very diverse and important algal lineage in nature. And a third, less well known and less abundant, but no less interesting, are the glaucophyte or glaucocystophyte algae. So three lines, and we believe that they all stem from the same primary endosymbiotic event. So next slide. Oops, could we advance? Yep. So I'll present a couple of different lines of evidence for the primary endosymbiotic origin of plastids being a singularity, and we'll see that there are some things that we know and some things that we do not know. Plastids have a genome, and biologists, as we've learned, have been sequencing and studying those the genes that are remaining in the plastid, and you can build phylogenetic trees and compare them to bacterial homologs, and there is no shadow of a doubt now that plastids are of cyanobacterial ancestry. That's also supported again by biochemical the biochemical and physiological properties of those two entities, that is oxygenic photosynthesis. What we don't yet know is the exact lineage of cyanobacteria to which modern day plastids are most closely related to. And this is a little cartoon from a few years ago. It's just a highly stylized picture of different types of cyanobacteria, from single-celled organisms on the left to more complex branching forms on the right. And depending on the data sets that have been used and the type of analyses done and the organisms that are included, over the years, people have seen plastid sequences linked to various branches within the cyanobacterial tree. So I would view this as still an open question in the field of early plastid evolution. So to recap, cyanobacteria are clearly the predecessors of plastids, but we don't know whether there is any modern day cyanobacterial group that is a specific ally of chloroplasts. Next slide. Another line of evidence that speaks to the singularity of plastid evolution is the nuts and bolts of protein import. And by that I mean when you look at the machinery that sits in the outer two membranes of a chloroplast, which is cartooned there, biochemists and molecular biologists have done a lot of detective work to figure out all of the bits and pieces of that protein import machinery. And we'll talk more about that in other contexts, but I think you will know that most of the proteins that make a chloroplast function are actually not encoded in the organellar genome. They are encoded in the nuclear genome of the plant or the algal cell, and those proteins are translated on cytosolic ribosomes and imported. Well, there's a very complex array of proteins that import machinery, and it's cartooned there for green algae in plants and also for red algae. And it's comprised of what are called tox and ticks. So these are translocon machineries, and the O and the I stand for outer and inner chloroplast membranes, respectively. Well, so when you do a detailed analysis of this, you'll learn a couple of things. Many of these ticks have homologs in cyanobacteria, so this speaks even more strongly to the fact that chloroplasts come from cyanobacteria. But also what you'll find is that there are, in fact, some algae with some of these protein import components still physically, the genes are still encoded in organellar DNA. So that speaks to the fact, again, the cyanobacterial connection to chloroplasts. But when it comes to the singularity, what we find is that if you look at green plants, an algae, the red algae, and the glaucophytes, which are not depicted here, you find that they share the same or very similar suite of proteins involved in plastid protein import. Okay, and the idea here is that that tick-tock translocon evolved on one occasion in a common ancestor of the three primary lines, and then has been kind of tinkered with over evolutionary time as they those three lineages diversified. Okay, next slide. Now, if we look at plastids and cyanobacterial genomes, we can learn some interesting things about the molecular mechanisms and events that transpired during that phase of time when a cyanobacterium was converted into a fully integrated plastid. This cartoon on the left is from Bill Martin and Jeremy Timmis, and what it shows you, it's rather complicated, so we'll just talk our way through it. You have to look at this diagram in 3 dimensions. So the height of each of those bars corresponds to the number of genes, and so on the extreme right-hand side of that, those are the representative number of cyanobacteria. So that's the 3 very high bars. Okay, and then all of the other bars are various forms of algae and plants. And you can see that their bars are much, much, much lower than those of the cyanobacteria. So all that is to say that there is a massive reduction in the size of the genomes of modern day plants and algae. So they all have a genome that is much, much, much reduced compared to any known free-living cyanobacterium. So genome reduction was a huge part of this process of converting an endosymbiont into an organelle. As it turns out, red algae, those that have been sequenced, have the largest and the most gene-rich plastid genomes that have been analyzed. And the different colors on the graph on the left correspond to functional categories. I won't dwell on this point, but the message here is that most of the coding capacity of plastid genomes revolves around proteins involved in gene expression, in particular translation, and as you might expect, photosynthesis. So That is to say, when you look at the green and yellow portions of those bars, they represent a much higher fraction of the algae than they do of the cyanobacteria on the right-hand side. And just to show a picture, for those of you who don't think about this very often, on the right-hand side is what I've called a typical plastid genome. There really is no such thing. That there are lots of different sizes and gene densities and so forth, but this is a prototypical one from Nephroselmis, so from a green alga, and that genome is 200,000 base pairs, approximately. And, you know, we're talking about 150 genes tops. So that's a fairly good-sized chloroplast DNA. There are much smaller ones than that. But again, just to emphasize the point, that's 200,000 base pairs, whereas modern-day cyanobacteria have, you know, several million base pairs of DNA and would have well over 1,000 genes. So again, emphasizing this massive reduction. Okay, next slide. So what I've been trying to convince you of is the fact that plastids evolved only once in the history of eukaryotic life. And that's sort of true, but in fact, the story is a little bit more complicated than that. And so I'm going to finish off by telling you about some interesting organelles inside an amoeba called Paulinella. And this is, on the left, the diagram is it's a hand-drawn diagram by someone named Robert Lauterborn, a German biologist, and you can see the era that he lived in there. In the late 1800s, he described this organism called Paulinella chromatophora. And on the right is an actual picture, a modern picture of light micrograph of the organism. And so you can see that Lauterborn drew a pretty accurate representation. It has two green sausage-shaped entities within inside the cell. Next slide, please. And Lauterborn was struck by the similarities between those sausage-shaped entities, the green sausages, and what he knew to be, at the time, had been described as cyanobacteria. So this is a quote from him in 1895. Either the blue-green structures represent algae from the division Cyanophyceae, so cyanobacteria, which are taken up as food, or they are independent organisms that live within the rhizopod or the amoeba in an intimate symbiosis. And it's even possible that they are integral components, real organs of the rhizopod cell biology. And this is a fascinating quote when you contextualize this, because it was written in 1895, which is a full 10 years before the paper by Merezhkovsky that is today held to be the first explicit articulation of symbiotic theory for chloroplasts. So next slide. So this is one of these examples where an interesting paper is largely ignored for many, many years in science. There are a few papers here and there between 1920 and the 90s, but very, very few of them. And it wasn't until 1990 in the lab of Michael Melkonian, a German biologist, who saw, with a student, saw Paulinella in natural field samples, and his student succeeded in producing a culture, establishing a culture of P. chromatophora in the lab. And Melkonian's group were the very first to obtain molecular data from those green chromatophores and the host organism, and it told a fascinating tale. Next slide. when these biologists actually analyzed the molecular data, that the sequences that are inside the chromatophores of Paulinella, they found that actually those green sausage-shaped entities are not related to chloroplasts at all. They are cyanobacterial, to be clear, and they carry out oxygenic photosynthesis, but they are from a distinct line. So you can see a cluster of 23 plastid sequences representing quote-unquote normal or canonical plastids up in the beta cyanobacteria, and Paulinella down in the bottom in the alpha cyanobacterial clade, most closely related to what are called Synechococcus and Prochlorococcus cyanos. Next slide. And as time went on, eventually people were able to sequence the complete chromatophore genome of this organism, Paulinella. And I hope you can see some of the numbers in the table here. The chromatophore genome is just over 1 million base pairs in size. So again, that's way, way bigger than any chloroplast genome or plastid genome from diverse plants and algae, and it had more than 800 genes. Okay, so much larger and much more gene rich. And yet, when you compare it to its closest cyanobacterial relative, which is a Synechococcus strain, 5701, that genome is 3 million base pairs and has, just over 3,300 genes. Okay, so this tells the story that these chromatophores are of endosymbiotic origin, clearly, but it is an independent origin from the typical plastids that we think of in plants and algae. And it is in the process of active reduction. Okay, the genome of the chromatophore has got lots of different pseudogenes in it, so broken genes. And this is an indication that it is undergoing reductive evolution, so the removal of genes that are no longer required for intracellular life. Now, if you do a molecular clock analysis to try and date the age of this endosymbiosis, researchers have found that it's on the order of 50 or 60 million years ago, which is very, very recent, of course, in evolutionary time relative to the origin of plastids, which is probably on the order of, depending on who you talk to, 800 to 1,200 million years ago. Next slide. Now, Paulinella has been a fascinating story to follow over the past 15 years or so, in part because there's been a very intense debate about what it is and what it is not. Should we use the word endosymbiont, or should we use the word organelle to describe it? And there has been arguments made on both sides of that debate. But what people have demonstrated, researchers, is that genes have moved clearly from the chromatophore to the nuclear genome of the amoeba. And that's pictured in the bottom right of this cartoon. And there is protein import into the chromatophore. So proteins that are the products of nuclear genes are synthesized on cytosolic ribosomes and imported back into the organelle. Next slide. And a real champion in this field in recent years is someone named Eva Novak. She has her own lab now in Düsseldorf in Germany. And this is in reference to a paper published in 2017. And her team used a proteomics approach to identify the protein components of these chromatophores. And so in the pictures on the left, you can see they basically popped open the cells and purified the chromatophores and carried out proteomic investigations. And because they had both nuclear and chromatophore genome data, they were able to show very clearly that there's at least 400 proteins that are nucleus encoded but are targeted to the chromatophore. And in fact, they use a very similar biochemical mechanism for making that protein import happen. That is the presence of N-terminal extensions on the front end of these targeted proteins. So this is a really interesting case. These words organelle and endosymbiont are not as easy to apply as they might seem at first glance. And in the case of Paulinella, it really does seem that the term organelle in its strictest sense applies. And by strictest sense, I mean there is both evidence for gene transfer, endosymbiotic gene transfer from the chromatophore to the nucleus of the cell, and also a dedicated protein targeting machinery has evolved to bring back proteins to make the chromatophore function. So whether we would want to call this organelle a plastid or not is essentially a matter of taste. Surely it is a photosynthetic, fully integrated organelle, but it is not of the same origin as the plastids that are found in all other known plants and algae. So it's a really interesting case, and there's going to be more exciting discoveries, I think, to come from the study of this system. Next slide. So photosynthesis is patchy. This is a segue into the second part of the talk after the break. I've told you, you can see the green arrow in the middle of this complicated diagram is pointing to the putative primary origin of plastids, giving rise to red, green, and glaucophyte algae. Up towards the top right is Paulinella, the organism that I've just told you about. It has an independent photosynthetic apparatus, the chromatophores. And then all the other colors on this tree are lineages that do not neatly fall into this category of primary endosymbiosis. And so that's going to be the second part of the talk, to explore how it is that plastids and photosynthesis has come to be spread far and wide across the eukaryotic tree. So next slide. And with that, I'll stop now. And thank you for your attention thus far. This is a shameless plug for a book that I wrote on this topic called 1 + 1 = 1. It's not been translated into Portuguese yet, but it is available in English, Spanish, and Czech. So if any of those three are of interest to you, I'd encourage you to check it out. It recounts some of the history that I've talked about and also modern perspectives on chloroplast diversity and symbiosis. So thanks very much. I'll stop, and I'd be happy to entertain any questions before the break.
Alguma pergunta, pessoal? Enquanto vocês fazem aí, eu queria fazer uma pergunta. I would like to make a question while the people think about things. How can we discover what lineage are more related to the plastids? What is missing until now?
Okay. You're talking specifically about cyanobacteria and so forth. Yeah, that's so the question is, what are we missing? You know, in a perfect world, if we really wanted to nail down where chloroplasts come from, what would we have to do? Or what would we have to have? And there's some known unknowns and unknown unknowns. And I think one aspect of this is the discovery aspect, which is to what extent are there novel forms of cyanobacteria out there in nature that have not yet been sampled that we can discover and sequence their genomes and then compare them to plastids and hopefully make meaningful connections. Now, I didn't mention it at that point of the talk, but one example is these Gloio margarita, these bacteria, cyanobacteria, on one of the slides I showed. There's a recent paper that showed a specific connection between modern plastids and that specific line of cyanobacteria. So that those cyanos weren't discovered until relatively recently. So, you know, it may sound like a bit of a stretch to say, well, we just have to wait around for new organisms to be discovered. But in fact, you know, that happens fairly regularly. So we might get lucky in that regard. So the less positive aspect of this question is more of a methodological one. And it has to do with the tools that are used to analyze the sequence data. And so I alluded to the fact that this tree building approach, you know, building phylogenies of plastid sequences and comparing them to cyanobacterial sequences, that's a tricky business. There's not that many genes left in plastid genomes, you know, at most about 200, and you can probably only analyze about 100 of those tops for various reasons. Either they don't, you know, they might not have good representation within cyanos or so forth. So, you know, the canonical origin of chloroplasts is a very ancient event. You know, we're talking, you know, pushing 1 billion years. So we are asking a lot of our molecular phylogenetic tools to be able to extract signal from DNA and protein sequence data. And by that I mean this is how different teams, different groups of researchers can use different tools and methods to analyze the same data and come up with different answers. So, oh, you know, in one analysis, plastids branch up over with these cyanobacteria, and then in another analysis, it's over here. It's telling you that we have very imperfect tools. We don't fully understand the, you know, sequence, the mechanistic approaches that we need to understand properly to extract that signal. So the people will always be developing more realistic tools of sequence evolution, and perhaps we will hone in on some answers that we can be more confident in. But I think it's just one of those grand challenges in biology. Some of the most interesting questions in evolutionary biology are also, you know, by definition, some of the most difficult ones. And so, you know, without a time machine, you know, we're left analyzing the sequences of modern organisms, and that's the way it is. Does that help?
Yeah, good. I forgot my microphone. Sorry.
Okay.
Another question from Watson.
Okay. That's a good question about chlorophyll evolution regarding chlorophyll B and C. Were they evolved independently? Ooh, can you make it come up again? I didn't finish. Or is there a straight ancestry line among them? What are the pieces of evidence? So chlorophylls B and C. I'm not a pigment specialist, so you can take my answer with a grain of salt. But what I can tell you is that those pigments, you know, each of those pigments has got multiple forms, you know, so there's chlorophyll C 1 and 2, and I'm assuming in B there's slight variations on that theme. And each of those pigments, you know, they've got different biochemical pathways and enzymes that are used for their production. So all that is to say that it's complicated. I don't specifically know of any connection between B and C. You know, we've also got chlorophyll A to contend with, and my understanding of the literature is that the pathways are kind of, there's enough flexibility within those pathways that it's not very clear to draw a direct connection between the various pigments. That's really the best I can say in terms of an answer, but I don't pretend to be a specialist in that aspect of chloroplast biology. So I hope that you get a better answer from some of the other instructors. But it's very complicated, and I'm told it's actually easier than one might think to use similar sorts of enzymes to produce different types of chlorophylls. So then that leads the question, with enough evolutionary time, you can tinker with the system and have a pigment that looks similar to another pigment, but it's produced using a different set of enzymes. That's the best I can say. Thank you for the question.
We have another question here. Maybe you can try to answer.
Where can we place the Xanthophyceae heterocontophyta in the evolution? So Xanthophyceae within heteroconts. So you don't mean heteroconts as a whole? I'm assuming you're referring to xanthophytes in particular. And I don't have a good answer to that question, other than to say that they belong. We might look at some trees in the second part of my talk that would more fully address that, but they are very clearly in the line, a monophyletic lineage of chlorophyll, AC pigmented plastids, along with diatoms and all of the other algae that we know and love. I don't have a specific answer to that particular question. I'm sorry about that.
Okay. The next question is divided in two parts. I will try to put the first part here.
Okay. Okay. Thank you for your question. It referred to the idea that there is an independent origin of the green algae and their land plant descendants and the red algae. And I think Wellerton, is that accurate description of that question? Could you put it up just so I know? The green lineage evolved with a cyanobacteria, while the red line descended from a different. So yes, that is widely believed to be wrong. The evidence, the molecular evidence, the structure of the genomes of red algae and green algae in land plants, and indeed the glaucophyte algae, the third lineage. If you look at the data from all three of those and ask the question, does it speak to one origin in their common ancestor, or two or more separate origins from different cyanobacteria? The answer is the former. That is, green algae and land plants, red algae, and glaucophyte algae. All of those three lines evolved. The plastids in those lineages evolved in their common ancestor, and their pigmentation diversified over time. And again, the evidence for this, the chromatophore example that I presented is an interesting case for comparison, because we can make predictions about the phylogenies that would come from cyanobacterial genes and plastid genes in the context, in a broad analysis. And also when we look at the protein import machinery, it's essentially the same in terms of its core componentry in all three lines. That is, there's very strong evidence for common ancestry of the plastids in all those lineages. Now, I'm not to say that 100% of the biologists in the field would agree with what I just said, but I would say 99.5% of them would. There are some papers going back to the mid-90s and around the 2000s by someone named John Stiller, who I admire greatly. He's been a real advocate of looking at alternate scenarios for plastid evolution. And so you will find papers out there that have proposed independent origins of red algae and the green algae. I'm not sure what his current perspective is on that at the moment, but the consensus, if I were to summarize it, it would be single origin. So thank you for the question. Okay, what do I think of this 2019 hypothesis on the origin of photosynthesis by giant viruses, horizontal gene transfer? I do know that paper. I guess I'll be honest and say I'm not quite sure what giant viruses have to do with it. There are some interesting connections there. But I honestly can't. It's a preprint. I think it might actually be published now. But I personally would not endorse that particular idea. I think giant viruses are a talk in and of themselves in terms of the way that they acquire genes and spread genes in eukaryotes. And there is some interesting data from Paulinella to do with viruses, but what that actually has to do with the origin of photosynthesis in Paulinella, I confess I'm not sure. So thank you.
Yesterday, we saw with Dr. Watson that is very common horizontal transference of genes in cyanobacteria. How can this fact influence the know that we have about the relationships about chloroplasts and the cyanobacteria?
Yeah, that's an excellent. So you're referring to the talk you had yesterday?
Yeah.
I wish I could have heard that, but that individual is quite right. Cyanobacteria are like many bacterial lineages, very dynamic in terms of their gene content, driven by lateral or horizontal gene transfer. That's absolutely true, and you're quite right to raise concerns about if that much gene transfer is taking place. How does that impact our ability to infer deep questions about chloroplast evolution? And William Martin in Düsseldorf and his colleagues, their group have been pointing this out and making very compelling cases for the reasons why we need to be cautious in our interpretations of such trees. So I think the idea is that if, whenever we do an analysis that includes chloroplast sequences and cyanobacterial sequences, we're doing comparing very anciently diverged sequences. And it's not as though the cyanobacterial sequences have not been changing since the chloroplast endosymbiosis. All those lineages have diversified and they've been swapping genes along the lines of what you just described. That has been ongoing. And so anytime we do an analysis where we compare chloroplast sequences to those of extant, modern day organisms, we have to accept the fact that those genomes do not represent what cyanobacteria looked like at the time of the endosymbiotic engulfment. That's a real problem, and it's one that we need to take seriously, for sure. I'm going to talk a little bit more about lateral gene transfer in the second part of the lecture, but it's an excellent point.
Okay. Then can we make a break now?
Mm-hmm. Yeah.
We can return what time?
Well, we can come back. It's up to me, is it?
Yeah.
We could. I guess we're a tiny bit behind. I guess if we want to give people enough time for a proper break, we could come back at 11:05. That would give people, I think, 20 minutes by my watch. Okay. Does that seem acceptable?
Yeah.
We'll start at 11:05.
Ok. Então nós voltamos daqui a pouco, pessoal. Olá, pessoal. Vamos voltando. Não se esqueçam de assinar a lista de presença, tá bom? Fiquem atentos aí para não perderem a oportunidade. Can we start again, John?
I'm ready if you are.
Yeah. Here is the presentation. What? Yeah.
Well, so that's the old file. I've emailed you the second slide deck, Valentin.
Okay. Só um momentinho, pessoal.
Yes, there we are, part 2. Okay, thank you. Well, thanks for coming back, people. It's great to carry on here. Next slide. So we talked about this figure already. This is, just to refresh your memory, the primary endosymbiotic origin of plastids. Next slide. What I want to do now is to dig a little bit deeper into the mechanisms that accompany that kind of an endosymbiotic event, and that's shown in this cartoon. So on the right is a generic primary plastid-bearing cell. So it could be a red alga, it could be a green alga, or a plant cell. It's just generic in that sense. And that modern-day cell is the derivative of an ancient primary endosymbiotic event, and that's cartooned on the left. Now, we don't know exactly the nature of the cyanobacterium that was engulfed by the non-photosynthetic eukaryote, as we spoke about in the first part of the lecture. We're limited by the organisms that exist and that we can study, but we can infer from modern-day cyanos that endosymbiont must have had between 1,500 and perhaps even 5,000 or more protein genes. And during the course of evolution, as red, green, and glaucophyte algae diversified, various things happened. And what are those things? Well, there was undoubtedly an extensive amount of gene transfer. So we can think about that as follows. If we look at the coding capacity of modern-day plastid genomes, they range between about 70 and 200 or so genes. Okay, give or take. Now, if we ask a plant biochemist who does proteomics how many proteins are in, for example, the Arabidopsis chloroplast, they will tell you it's several thousand. So, so the question becomes, what gives? How can it be that the genome only has at most 200 genes, but the proteome of the organelle is in the thousands of proteins? And the answer to that is endosymbiotic gene transfer. So the arrow moving from left to right indicates the movement of genes from one compartment to the other. And then the protein products of those genes are re-imported back into the organelle. So, you know, at most 5% of the proteins that make a plastid function are actually physically, you know, the genes are physically located on plastid DNA. Most of them come from the host part of the equation. Now, those other arrows pointing in different directions are meant to illustrate an interesting phenomenon, which is that it turns out if you carry out a comparative genomic investigation, and William Martin and various other research teams have done this, and you ask the question, in plants and algae, where do all of the cyanobacterial derived nuclear genes, where do their gene products function in the cell? You find out that many of them, of course, function in the plastid, as we would expect and as is shown in the diagram, but many others too are targeted to different locations. So we have 3 other arrows, one going to the mitochondrion, MT, one going to the cytoplasm, and another going to the secretory pathway. So all this is to say that cyanobacterial proteins contribute to more of the molecular and molecular biology and biochemistry of modern day plant and algal cells than just the plastid itself. Okay, and this is, as we'll see, a recurring theme when you dig deep into the details. There is a disconnect between the origin of a particular protein, in this particular case, cyanobacterial proteins, and the actual compartment in which they function. Okay, so next slide. Now, when we were talking about photosynthesis being patchy before the break, what I meant by that was that algal groups are distributed across the eukaryotic tree in interesting ways, and that is because of this phenomenon called secondary endosymbiosis, and that's the cartoon on the left. Secondary endosymbiosis, unlike primary endosymbiosis, involves a eukaryotic host and a eukaryotic endosymbiont. So you have one eukaryote engulfing another eukaryote, and the engulfed eukaryote is itself a primary plastid-bearing alga. Okay, now there's a list of algal taxa there, and we'll talk about some, but not all of these. I suspect they'll come up at different parts of this course, but suffice to say that there are examples where the endosymbiont was a red alga, and those are the ones listed in the red box, and then there are others that are where the endosymbiont was a green alga, and those are also listed. One of the things that all of these, what are called secondary plastids, have in common is 1 or 2 extra membranes around the plastid. Okay, and that, I might or might not have mentioned it, primary plastids are characterized by having 2 plastid membranes. So the organisms listed there have either 3 or 4 membranes surrounding their photosynthetic organelles. Now there's even something called tertiary endosymbiosis, which is common in dinoflagellate algae. I'm not going to talk about it extensively today, but it, as the name suggests, is yet another round of endosymbiosis where you've got a secondary plastid-bearing organism engulfing some kind of alga that itself already has a secondary plastid. So things get even more complicated. We have these nested relationships of symbioses. Now back to the membranes. So we'll jump to the next slide. Now this, it was a long time before biologists figured out that secondary endosymbiosis was a thing. And one of the first people to do it, to put the pieces together was a biologist named Sarah Gibbs. Now, she described it as a bolt from the blue. Now, what you're looking at there on the right is a light micrograph of a euglenid, euglena. Many of you will be familiar with that organism. And the story goes that Dr. Gibbs was reviewing a paper about, I think it was mitosis or something, cell biology of euglena, and she was trying to reconcile the fact that the pigmentation of the euglena chloroplast, that is chlorophylls A and B, was very much, it was essentially the same as what you see in green algae and land plants, but the broader biology of the euglena cell was utterly unlike a green alga. And it dawned on her at that time that, and I can read the quote here, ultrastructurally, euglena could not be more unlike green algae. Out of the blue, I suddenly realized that euglena was not related to green algae at all. It just ate them for dinner. The moment I thought of it, I was ecstatic. I knew it had to be true. It explained too many things not to be true. Now, what did it explain? Well, Sarah Gibbs had been studying chloroplast biology in diverse algal forms for many years. So all of the heteroconts, haptophytes, cryptophytes, and so forth, she knew what their chloroplast envelopes looked like, and she knew that they all had additional membranes surrounding them that at that time really had no explanation. And so it was in the 1980s that she started to put the pieces together and recognize that, in fact, these organisms were the product of eukaryote-eukaryote symbioses. Okay, next slide. And you will be familiar, I think, with many of the organisms that have acquired photosynthesis in this fashion, so I won't belabor this point. We've got things like dinoflagellates, so very common bloom-forming organisms, heteroconts, or stromatophiles if you prefer, a very, very diverse group of organisms, many of which are photosynthetic and plastid bearing, and many others that are not. So, things like giant kelp in the ocean, those organisms superficially to someone who's not a specialist would think, well, those are, they're related to plants, to green plants, but of course nothing could be further from the truth. They're actually more closely related to the single-cell diatoms that are floating around in the sea. And even organisms like malaria parasites. So on the right is a couple of pictures of Plasmodium falciparum, which is the malaria parasite. It belongs to the apicomplexans, a group of predominantly parasitic protists. And these organisms, in particular Plasmodium, is not photosynthetic, but nevertheless it has a plastid that is the location of essential biochemical processes that explain why this plastid still persists, even though the organism has long since lost the ability to photosynthesize. Okay, next slide. And again, I've shown you this picture already, but haptophytes, another very, very important lineage. E. hux, Emiliania huxleyi, would be probably the most famous. Massive bloom formers in the world's oceans. The White Cliffs of Dover are comprised, at least in part, by the coccoliths, as some groups of them are referred to as. Next slide. Now, the analogy that you will often hear when it comes to thinking about secondary and tertiary endosymbiosis is this idea of Russian nesting dolls. So I think we've all seen these kinds of things. It's the idea that you can make a set of nested dolls, and the head of each doll comes off, and you can put the the smaller doll inside a bigger doll, and then that bigger doll can go inside a still bigger doll, and so forth. That's an interesting way of thinking about cells inside other cells. The analogy kind of breaks down, though, when we start to think about the nuts and bolts of what's going on with secondary endosymbioses. Next slide. And that is because, as we discussed earlier, there is this phenomenon of endosymbiotic gene transfer. So this is a cartoon which shows these what I call waves of EGT, endosymbiotic gene transfer. So each time you have an endosymbiotic event, if it persists long enough, it is accompanied by the movement of genetic material from the endosymbiont to the host. And now initially, in the case of primary plastids, that endosymbiont was a cyanobacterium, or CB as is depicted here. And in the context of secondary endosymbiosis, you've got the movement of genetic material from the nucleus of the primary host, and that is labeled N1 on this diagram. Movement from that nucleus to the secondary nucleus, or N2. And with each of these waves, these nested sets of endosymbioses, again, if it persists long enough, the cells evolve protein import apparatuses. So those are shown in yellow. Okay, so the little cartoon chromosomes there are multicolored, and that's to illustrate the fact that they are mosaic. So the genes come from different locations, different organisms. They have spent time over evolutionary history in different genetic compartments. And on the furthest right side there, the nucleus of the secondary host is a mosaic of genes from a number of different sources. Now, what I have indicated on the bottom, that LGT, that stands for lateral gene transfer, and we spoke about this before the break. This is the idea that organisms can pick up genes from unrelated organisms by various mechanisms, and we know that this happens certainly in bacteria, and it happens at least to some extent in eukaryotes as well. And so when we combine EGT with LGT, what we end up with is a very, very complex picture of the evolutionary history of the collection of genes that are sitting in the secondary host nucleus. And I'm going to return to that point a little bit later on. Next slide, please. This is the patchy diagram I mentioned. I'm going to spend time, the next little block of slides relate to some of the work in my lab over the past 15 years or so, related to two lineages of algae. One is in the top left, and that's the red arrowhead pointing to the cryptophytes. And the second lineage is an unrelated lineage over on the right part of the tree called the chlorarachniophytes. And they're colored red and green for reasons that will become clear in a second. So next slide. The cryptophytes, if you've not heard of them, are a pretty diverse and ubiquitous group of algae. They live in both marine and freshwater environments, and brackish water too. Not particularly abundant, but nevertheless important. They are interesting from an evolutionary and a cell biological perspective for the fact that they have plastids that are surrounded by four membranes, as I mentioned earlier. And they also have what's called a nucleomorph. And I'm going to spend some time explaining what that nucleomorph is. In essence, that's considered by many to be what we call the smoking gun of secondary endosymbiosis. So cryptophytes have two nuclei. They have a host nucleus, which is much larger and much richer in terms of its genetic complement. And then they also have a residual endosymbiotically derived nucleomorph, which I'll explain in a moment. Next slide. The other lineage of nucleomorph-bearing algae are the chlorarachniophytes. Now, these are predominantly marine algae, somewhat less abundant than cryptophytes. And you can see that they're green, so they have chlorophyll AB pigmented plastids. That's where the chlora part of the name comes from. And the arachnio part, that refers to spider. So the chlorarachniophytes, I guess loosely translated, would be green spider. And you can see from the images that I have presented there on the left, they form these, it's basically an amoeba-like host with these spider-like projections coming off the cell. So that not all chlorarachniophytes look like that, but many of them do. And the TEM in the bottom right-hand corner illustrates, is meant to show you the two different nuclei. So NU being the host nucleus, and then NM being the nucleomorph. And like the cryptophytes, the chlorarachniophytes have two membranes around their plastids. So, sorry, two outer membranes, making a total of 4. And the nucleomorph sits between the two membranes, two pairs, the inner and the outer pairs. So chlorarachniophytes have a green algal-derived secondary plastid, and the cryptophytes have a red algal-derived secondary plastid. Next slide, please. And what that means is nucleomorphs evolved twice. So different origins of the algae that gave them photosynthesis, and this independent origin presents us with the opportunity to carry out some comparative biology to try and understand this phenomenon of secondary endosymbiosis and how it's contributed to the two different types of cells, two different hosts, and two different flavors of algae, red algae on the left and greens on the right. Next slide. And so we've been working on this for, well, almost 20 years now, I guess, in my group since 2003, have worked on comparative nucleomorph genomics and tried to understand these waves of reductive evolution. And that's what those little well, they're sort of like arrowheads, I guess. So we know that there is intense bursts of genome reduction that have happened early in the secondary endosymbiosis. We don't yet have the organisms available to make much sense of what happened in the ancient past, but we're studying modern day representatives of these two lineages, cryptophytes, or cryptomonads as they're sometimes called, and the chlorarachniophytes in a sort of a compare and contrast perspective. Next slide. So I'll just tell you a little bit about nucleomorph genomes, if you've never heard this story, but because it's quite interesting. The first nucleomorph genome to ever be sequenced was that of a cryptophyte. It was published in 2001. That's a cartoon that you can see of the cell. The genome, the nucleomorph genome, is 550,000 base pairs, or kilobase pairs, 465 genes predicted, and there are 3 chromosomes. So the genome is broke up into 3 different chromosomes and is about 500,000 base pairs in size. So this is vastly smaller than any known red algal genome. Okay, so very, very reduced. Next slide. Jeff McFadden's group in Melbourne, Australia, some years later followed up with the first chlorarachniophyte nucleomorph genome. it was even smaller. This particular one is 370,000 base pairs, 330 genes. So again, very, very reduced relative to known green algae. So an intense amount of reductive evolution has taken place. One of the oddities that was clear when these two genomes, nucleomorph genomes, were sequenced and compared was the fact that they both are comprised of 3 chromosomes. And that's the cartoon on the right-hand side. And they also have their ribosomal RNA operons located in a subtelomeric region, so on the ends of the chromosomes. Now again, this is a bit unexpected and surprising because these are of independent origin. So again, chlorachnophyte nucleomorphs are from green algae, the cryptophyte ones are from red algae, and they have independently converged or been shrunken down to this three chromosome state, in which they've put their rDNAs on the chromosome ends. And we've published a number of nucleomorph genomes, additional ones, ourselves over the years, and there's really no exceptions to this particular rule, and so no one actually understands the meaning of why there is 3 chromosomes in both lineages. Next slide. If you're wondering what it is that nucleomorphs encode, with the few hundred protein genes that they have, the answer is little to nothing in the realm of primary metabolism. So they don't encode much of the core metabolism that makes these algal cells tick. Those metabolic enzymes are encoded in the host nuclear genome. They essentially contain a residual set of housekeeping genes, sort of transcription, translation, protein folding, those kinds of cellular processes, and a small number of plastid proteins, so on the order of 15 to 30 or so. So a very, very incomplete set of genes. Next slide. And if I were to sort of summarize 3 sort of take-home messages about what you might want to remember about nucleomorph genomes, they're extremely small, as I said. All of them have 3 chromosomes that have been looked at to date. The genomes themselves, if you're not someone who works in this particular area, the genes are very, very dense. So I'm talking about around 1 gene per 1,000 base pairs of sequence, and that is very much like a bacterial or an archaeal genome arrangement. They even have overlapping genes, which is something that's not very common in nuclear genomes. And they're also very weird insofar as their base composition is quite biased. So they're on the order of about 75% AT, so 25% GC. Very compositionally biased and fast evolving sequences, which is interesting, but also makes them quite difficult to study from an analytical perspective. So this phenomenon of reductive evolution, the nucleomorphs, as I've said, they are eukaryotic in origin. They are nuclear genomes, but in terms of their properties, how the genes are structured and their sizes, they're more akin to like a chloroplast or a mitochondrial genome in terms of the gene density and their base composition. So this phenomenon of reductive evolution that you often see in organelles and bacterial endosymbionts, we see the same hallmarks of that in the nucleomorph genomes that have been studied. So it's a recurring theme in evolution. When an organism comes to reside inside another organism, interesting things happen to the genome. It shrinks, it becomes more compact, and it becomes more compositionally biased in terms of its nucleotides. Next slide. So one of the longstanding questions in this field is, why do nucleomorphs persist? And we established a collaboration with various international partners, next slide, to address this question. And it revolved around the sequencing of the host nuclear genome of a representative cryptophyte and a representative chlorarachniophyte. And this is just a close-up of the cell biology, a cartoon that I drew of a cryptophyte cell. And I want to just emphasize the complexity of these cells. I told you about the nucleomorph genome really having fewer than 500 genes. Most of the genes in these organisms are found in the nuclear genome, in the NU compartment there. And until we embarked on this project, there was basically no data for the nuclear genomes of either cryptophytes or chlorarachniophytes, and so we wanted to sequence a nuclear genome to better understand the biology of these cells. And one particular area of interest is this, what's called the periplastidial compartment, and I've highlighted it red in this diagram. And what that compartment is, that's essentially the cytoplasm of the engulfed algal cell. So again, because it's a secondary endosymbiosis, the alga brings with it as an endosymbiont a nucleus, which eventually becomes a nucleomorph, and it also has a residual cytoplasm. So organisms like this are among the most complex in terms of their cell biology and molecular biology of any cells known, because they have two distinct cytoplasmic compartments where you have transcription and translation taking place, and you have 4 distinct DNA containing compartments. And so in order to kind of put the whole puzzle together, we needed to have the last piece, which was the nuclear genome. Next slide. This is a picture of some of the researchers who were involved in this project. It was funded by the Joint Genome Institute in California. And we met in September 2011 to start to craft the paper where we describe those nuclear genomes. And I'm going to tell you a little bit about some of the analyses that we did and some of the things that we learned. Next slide. This is a cartoon of the cryptophyte, Giardia and Bigelowiella. So again, the red and green coloring is obvious, I hope. And I've put the numbers of the genes involved that were identified in the nuclear genomes. Both of them were less than about 100 megabase pairs in size. So relatively straightforward to sequence and predict genes. And you can see from the numbers, 24, close to 25,000 genes in the nucleus of the cryptophyte on the left, and close to 2,200 for the chlorarachniophyte on the right. And next slide, please. We carried out a whole array of analyses on the suites of genes that we found in those nuclei. And part of what I'm going to tell you about today is this phenomenon of endosymbiotic gene transfer. So those arrows from nucleomorph to nucleus, that's depicting the movement of genes from one compartment to the other that took place at some point over evolutionary time in each of these lineages independently. And we identified 508 algal genes on the left and 353 on the right. And then we asked questions using bioinformatic tools about where the protein products of those genes function in the cell. And those are the arrows with the question mark. Do they make their way back to the plastid and the nucleomorph and its associated PPC, or do they end up in different places in the cell? perhaps the secretory pathway, or functioning in the cytoplasm. Next slide. What you see here is a pie chart summary of both, of a lot of work that went into this investigation, but I can summarize it by posing a question and then an answer. So again, of those 500 or so algal genes that we identified in Giardia, where do the protein products, where are they predicted to function? And the color coding are the different cellular compartments. And you can see that in Giardia, 66% of those were not predicted to function anywhere else in the cell other than the cytoplasm. 23% in the targeted back to the plastid, that's the green portion of the pie. And then other categories there are listed, mitochondrial, secretory, and so forth. And I think you can see qualitatively the picture is very similar in terms of the proportions between the cryptophyte on the left and the chloracniphyte on the right. So the question, where do these algal proteins function? The answer, simply put, is everywhere. Yes, we do see algal genes in the nuclear genomes of both cryptophytes and chloracniphytes targeted back to the plastid, but we also see them targeted to other compartments. And this is similar to the picture that that I described at the opening slide of the second talk here. That is to say, there is a disconnect between the protein and where it comes from evolutionarily, and the location of where it actually functions in the cell, which was quite surprising in many respects. Next slide. A related question revolves around whether EGT, endosymbiotic gene transfer, is still happening today in those two lineages, the cryptophyte Giardia and the chloracniphyte B-natans. Now, we addressed this question using fine-scale analyses of DNA sequences. So in the previous slide, I told you that we'd identified hundreds of algal-derived genes in the nuclear genomes. And that makes sense. We know that EGT has happened over the course of algal evolution in both cases. But here we wanted to ask the question in a more detailed way, which is there evidence at the DNA level of very, very recent transfer? And this is something that is seen in lots of different types of algal systems. For example, there are acronyms for these types of transfers. So NUMITES, NUMTS, that stands for mitochondrial DNA that is incorporated into the nuclear genome of an organism. And NUPET, NUPT, that stands for plastid DNA. And we were the first group to carry out an analysis of nucleomorph DNA transfers. And so NUNMs, that's the acronym we use for that. Now, to cut a long story short, we found a handful of examples of the NUMITES, so 13 and 9 instances in the case of Giardia and Bigelowiella, respectively. So we do find evidence of very, very recent transfer of mitochondrial DNA being transferred, but you can see from the large zeros there was no evidence of transfer of plastid or nucleomorph DNA, very recent transfers in either nuclear genome. So that was interesting. Next slide. And the reason that's interesting loops back to this overarching question of why do nucleomorphs persist? Now, I've told you already that this phenomenon of secondary endosymbiosis is widespread. It's been a driver of algal evolution, and all of the organisms listed on the right are known to have plastids surrounded by 3 or 4 membranes. That is, we know that they are derived from secondary endosymbiosis. But only 2 of those lineages, the ones with the ticks, actually retain the nucleomorph. So the question is, what is it about the biology of those lineages that has led to the persistence of that nucleomorph? And what is it about those other lineages that has led to the process of EGT going to completion? Now, a colleague of mine, Tom Cavalier-Smith, had a very nice quote in 2005. He said, "The optimum size for a nucleomorph genome is 0. And by that he means that, wouldn't it be simpler or easier for an algal cell of this type to just completely get rid of the nucleomorphs entirely? Why not move all of that DNA over to the host nucleus, and then you can get rid of the nucleomorph completely, and you don't have to have 2 different nuclear compartments where you have to maintain DNA replication, transcription and translation, and so forth." So it's clearly happened in haptophytes and heteroconts and other algae. Why hasn't it happened here? Next slide. Well, the answer, it dawned on us over time, relates to something called the limited transfer window hypothesis. And this was put forth by Chris Howe, Adrian Barbrook, and Saul Purton, UK researchers. And it is built on the premise that if you examine the underlying mechanics of EGT, that is the movement of DNA from an endosymbiont or an organelle to a nucleus, it is underlaid by organelle lysis. So that is to say the substrate, the material for organellar DNA to be transferred into a nuclear chromosome comes from the lysis of an organelle. And these authors postulated that there is a correlation between the number of organelles in a cell and the frequency of EGT. And that hypothesis was very clear in experimental work that I don't have time to articulate. The bottom line is that in tobacco, you can show that the transfer of plastid DNA to the nucleus is very, very frequent, and tobacco cells have hundreds of chloroplasts, or 100 or so, thereabouts. And in Chlamydomonas, essentially, you cannot detect in an experimental context any transfer at all. And the difference between clammy and tobacco and other organisms is that Chlamydomonas only has a single chloroplast per cell. And this analysis, or this idea, was followed up by David Smith, and that's a graph from a paper of his. And he showed that not only for plastids, but also for mitochondria, that you find many more examples of recent EGTs from plastids and mitochondria in organisms which have more organelles per cell than you do in the, and that's the polyplastidic and polymitochondrial part of the data, than you do find for the organisms that have a single plastid and or a single mitochondrion. Okay, so there's a very clear correlation in the data. If you only have one organelle, you see very little EGT. Next slide. And what does that have to do with cryptophytes and chloracnophytes? Well, from these cartoons I've been showing you, these organisms only have a single plastid nucleomorph complex per cell. And so our interpretation of the data that we are looking at is that essentially the reason that nucleomorphs persist in these 2 lines is that the genomes are frozen. They simply don't have the opportunity, because of the way the cells are organized, they just don't have the ability to transfer DNA to the nuclear genome at any appreciable frequency. So the host genomes are mosaics of genes from both eukaryotes, but that mosaicism is a product of some transfers that happened much longer time ago in evolutionary time. Okay, next slide. So hopefully I've convinced you that some of the ideas about why it is that algal endosymbiosis drives algal evolution, so these waves of gene transfer that I referred to, and the complexities of lateral gene transfer. Next slide. And it's led to this increasing appreciation of the fact that the nuclear genomes of extant eukaryotes are mosaics. Those nuclear genomes contain genes that are derived from by endosymbiotic gene transfer from the predecessors of mitochondria and chloroplasts on the left. But they can also contain genes that have come in from viruses. This is something that we're gaining an increasing appreciation of. And also from lateral gene transfer on the right. And so when you are analyzing the suites of genes that are in a newly sequenced algal genome, it can be very difficult to distinguish between genes, to pick one of these sources. Is it an endosymbiotically derived gene, or is it the product of lateral gene transfer? And in fact, all of these sorts of events are able to donate, at least to a certain extent, genes to eukaryotic nuclei. Next slide. Now, one of the ways, of course, we've learned about algal evolution is to study algae, but increasingly it's become clear that there is much to learn about the evolution of algae and their plastids by studying their non-photosynthetic relatives. And the heteroconts would be an example of that. I'm not going to talk about them specifically today, but in the case of the cryptophytes, with collaborators, we've studied an organism called Goniamonas, which is a relative of Goniamonads, and those are pictured on the left there. The cryptophytes, when you analyze their placement on the tree of eukaryotic life, you find that they're most closely related to a large diversity of heterotrophic plastid-lacking forms, Goniamonads, but also other protists, catablephrids and telanemids, for example. And those red arrowheads are depicting possible points at which the secondary plastid of cryptophytes could have come in. Traditional models have often placed that endosymbiotic event in very, very far back in evolutionary time, down at the very bottom. And this would mean that these heterotrophic forms had lost photosynthesis secondarily. And this was one of the reasons why we wanted to study Goniamonas in more detail. Next slide. So long story short, a former postdoc of mine, Unsu Kim, discovered a novel Goniamonad called Goniamonas avonlea from Prince Edward Island, just north of where I live. And with a group of people, we sequenced its genome. The long story short was we were looking specifically for the genetic legacy of past endosymbioses, whether there were algal genes in the nuclear genome that would perhaps betray its past life as a phototroph. The short answer is that we did not detect any that we were convinced by. Next slide. And this leads us to conclude that probably the cryptophyte plastid is of more recent ancestry than has been proposed in the past. That is, it probably came in a common ancestor in an organism of cryptophyte on the cryptophyte line after that line diverged from the goniomonads. So this is, again, an example where you can learn interesting things about the timing of endosymbiotic events on the eukaryotic tree by looking at the heterotrophs. Next slide. So let's loop back then to this puzzle of plastid evolution, as I call it. And I do see it as a puzzle because it's one of these questions where you can look at it from lots of different directions and collect bits and pieces of the puzzle and start to put them together. But until you have all the pieces in play, you can't be certain of what the actual outcome, the full picture of the puzzle is. And so the field of plastid evolution, when you think about eukaryotes as a whole, is still very uncertain in terms of the number of endosymbiotic events that have transpired. We know, or many people would agree with the idea that primary plastids evolved only once. And we know that there are red algal type secondary plastids in various positions on the tree, and there are also green algal ones. But we don't actually know how many of those are the product of secondary endosymbiotic events and how many of them might actually be tertiary events, as is seen quite frequently in the dinoflagellate lineage. And the real challenge in this field is comparing and contrasting different types of data. So you can imagine lots of different scientific evidence brought to bear to this question. We have ultrastructural data, so you can look at membranes and membrane topologies. You can look at pigmentation data of the plastid, and you can also use molecular phylogenetics of both host nuclear and nucleomorph and plastid genomes. But oftentimes those different types of data sets point to different conclusions. And so the real challenge for this field moving forward is to integrate and combine these different lines of evidence into a coherent picture of plastid evolution, but the field is certainly not there yet. Next slide. I want to end by with a quote from Charles Delwich from the University of Maryland. Now, Charles is the guy who's going to tell you about land plant evolution in, I think, next week or so. And I'm sure you'll enjoy that talk a lot. This is a quote from a paper that he wrote in 2004, and I love it because it illustrates how I think about genomes in algae and the complexities of the sequences that are contained in the genomes of modern day organisms. So the quote is, "The genome is like a palimpsest, an ancient recycled manuscript in which the traces of an earlier text can be discerned." And so that's a picture of a palimpsest on the left. If you've not heard that word before, that refers to these ancient texts where people wrote and rewrote on them, and sometimes in different directions, presumably to save precious paper or writing material. But there are imprints of past writings on that text, and if you try hard enough, you can actually discern what the earlier versions of the text actually were. So the quote carries on: "The current functions of an organism's genome represents the adaptations of the organism to the present environment, but these functions are superimposed on remnants of evolutionary history." So the genomes of modern day algae are mosaics, as I've said, and they've been written and rewritten multiple times as multiple rounds of endosymbioses have taken place. And the challenge is to be able to try and extract meaning from those different versions of that text. Next slide. So to bring this all to a close, in the field of algal and eukaryotic genomics, I like to make the reference to the field of prokaryotic comparative genomics. So about 20 years ago, technologically, it became possible to sequence prokaryotic genomes at high speed, high accuracy, and at low cost. And this led to a deluge of bacterial genomes and archaeal genomes. And there were lots of growing pains in around the 2000s where people were trying to make sense of the data that was observed in bacterial genomes. And one of the things was how much lateral gene transfer apparently was happening. And the field of eukaryotic comparative genomics, of course, is further behind because of the increased size and complexity of the nuclear genomes relative to prokaryotes. But now, of course, the technology is there that we're accumulating nuclear genomes at a prodigious rate, and we're coming up against some of those same questions. The relative importance of vertical versus horizontal inheritance. And in particular, in the algal realm, how is it that we detect and deal with these, what I call the footprints of endosymbiotic versus lateral gene transfer? We often have great difficulty telling the difference between them. And depending on where one lies on this spectrum, one can come to rather different conclusions about the evolutionary scenario that you're interested in studying. And there's also the question of, have all the lineages even been sampled? And it is the case that new algal lineages continue to be discovered. If we go to the next slide. This is a paper from some colleagues of mine in Japan. Two of these individuals are former trainees of mine. And this is the discovery of dinoflagellates, two different dinoflagellate lineages that have what appear to be nucleomorphs. So I've been telling you that there are only two algal lineages with nucleomorphs. But it turns out that in the dinoflagellates, there are these two lines now, and there is convincing microscopic and molecular data for the fact that these lineages have green algal derived plastids and nucleomorphs. There are no nucleomorph genomes yet available, but it's going to be very interesting to see those when they come out. So the next, 5 to 10 years, I think, are going to be a really exciting time for people who are in the business of trying to understand endosymbiosis and plastid evolution. So I'll end with the final slide, which is just to thank you and members of some of my team, past and present, and the funders. So I'll happily take any questions that you might have. Thank you.
Thank you very much for your presentation. It was very good to know about that. We have some questions that I will present for you here. Just a moment, please. Here.
What do I think about the red carpet theory to explain the mosaicism of genes found in some secondary lineages? For example, euglenids and chloracneophytes. Thank you for your question. That's a really interesting one. I'll unpack that a little bit by saying that the red carpet theory. So those two lineages that you mentioned there, euglenids and chloracneophytes, have green algal derived secondary plastids. But what has become abundantly clear, and is something that I didn't have time to explain fully today, is that if you look at the nuclear genomes of those two lineages, you will find many green algal derived genes, yes, but you will also find many genes and proteins that have affinity to red algae. Now, similarly, if you look at the cryptophytes and heteroconts, for example, and diatoms and things, you will find they have secondary plastids of red algal ancestry. So their nuclear genomes have many red algal type genes, but they also have green algal type genes. So I refer to this as red-green mosaicism. And that's how I tend to refer to it. So in both cases, there is a disconnect between the type of organelle that currently resides in the cell. In one case, a red algal type plastid, and then the nucleus of those cells has red and green genes. And then in the other case, you've got a green algal plastid and nucleomorph, and then in the nucleus, you've got red and green genes. So what's up with that? How do we explain that? And the red carpet hypothesis that you refer to there is in fact one of several different models out there to try and explain this red-green mosaicism. And I don't buy the red carpet model any more than I buy any other model. I think it's a perfectly reasonable hypothesis, and it's one of several that try to understand this mosaicism. But I don't think any of them is the sort of clear winner. Because I think, again, as I was saying in the talk, I don't think we fully understand until we have a more robust phylogenetic framework to place and pinpoint these lineages, we can't actually reconstruct the evolutionary history of the plastids. Some people would explain that mosaicism by saying, okay, maybe euglenids and the chlorarachniophytes, at some point in their ancestry, they had a red type secondary plastid, and then a green type came in and replaced it. The old one was lost, and that explains the mosaicism. I think that's possible in some cases, and it may ultimately explain one or two examples, but what we see is every single lineage you look at, you see the same mosaicism. And to my mind, we can't distinguish this plastid replacement scenario that I just described with lateral gene transfer. So that is, you don't have to infer an ancient plastid that was lost. You just have to have uptake of additional genes from different sources, in some cases red, in some cases green, and that can explain the pattern. So all this is to say, when you see the mosaicism, to my mind, there are multiple explanations for it, but I don't think the data in hand allow us to distinguish which is the better explanation. So I hope that helps.
Well, it seems that EGD is not simply an evolutionary fact, but also some kind of evolutionary advantage for the groups. I don't know. But we saw a lot of groups that have a lot of plastids in the cell,
yeah,
and also some derivative groups that lost the great number of plastids and retain just one. For example, some diatoms, some derivative diatoms have this. But would not be a contrast in this case, because the derivative group actually lost plastids and lost a lot of how can I say the capability to make EGTs, because the reduced number of plastids of organelles.
Yeah. Mm-hmm. Yeah, so you're talking specifically about plastid-bearing organisms. You're not talking about organisms that have lost plastids entirely. You're talking about the number of organelles per cell, right?
Yeah.
So I think that's interesting, and I don't think the limited transfer window hypothesis that I posited there, I don't think it explains everything, but I think it does a pretty good job. And one of the uncertainties there is that we can't know how many plastids was present, let's say, in the common ancestor of a particular heterocont lineage. We're looking at the tips of the trees. We're looking at the lineages that we have today, and we can ask questions. Okay, this one over here has got, they have dozens of plastids per cell. This one over there has only got one. That's what we see today, and we can make an inference between the organelle number and the number of the amount of EGT we see, but we can't know how it was further back in time. So I think that's why I like this idea of a window, because I think it kind of explains the variation that you can see in EGT over time. So, early on in an endosymbiotic event, I kind of think of it like, the window is wide open. Endosymbionts are being taken up regularly. They're being digested actively. And there is a lot of DNA that's raining down on that nuclear genome. And then, as the endosymbiont becomes more integrated into the host, and maybe the number of endosymbionts per cell goes down, then the window sort of starts to shut, right? And I think it's probably the case where there is a finite time where in the case of nucleomorphs, for example, if there's enough transfer, when the window is still open, then the nucleomorph can completely disappear, and before that window shuts, right? But if the window shuts before all the EGT takes place, then it can't easily go to completion. So, and I think it's also the case that windows can open, they can close and then open again. I mean, a lineage could diversify and end up having more organelles per cell, and then that transfer window can open again. So it's, we don't want to, I guess what I'm trying to say is you don't want to put too much stock in the tips of the tree, because you can never know exactly what the cells looked like further back in time. And a lot of those transfers, I guess I didn't really make this point clear, but when you do those sorts of fine scale analyses, almost all of those EGTs are meaningless in the fullness of time. So, in tobacco or in Arabidopsis, for example, when you look at the nuclear genomes, they're full of plastid and mitochondrial DNA. But that's not to say that those are functional genes. They still have perfectly good organellar genomes. It's just the system is dumping garbage DNA down on the nuclear genome. It lands there, and eventually it just drifts off. So almost all of it is dead on arrival, as we say. So it's a process that can happen over and over and over again if the window is open, and occasionally a gene will establish a function, and then the organellar genome that gene can be lost. But it's like a ratchet that can go over and over and over again. So it's a mistake to think of it as being optimal and successful all the time. I think almost all of the transfers are completely meaningless. If you look at the human genome is full of mitochondrial DNAs being integrated, even within the modern human population. Occasionally, they even cause diseases and so forth. But that's not to say that the human mitochondrial genome is getting smaller and smaller over time. It's just it's a product of the way that the cell biology is. Does that help?
Yeah, okay, man. We have another questions. I will try to make a resume of this, the main of this.
Is there a molecular explanation on how the genes are transferred between the plastid and the host, considering they are located in topologically different compartments? Yep, so that's a good question. The premise that underlies the answer is that organelle lysis is the starting point for all EGTs. That is to say, if you're a tobacco cell and you have 100 chloroplasts, let's say, in your cell, if one or two of those pop open, because of autophagy or something and are recycled. Occasionally, the DNA from those organelles can just make its way into the nucleus and can be integrated. Okay, so that's why that's the window open model, right? Organelles, there's many of them, so the cells won't die if one or two of them are lysed. So that's where the DNA comes from, and experimentally that's been shown. But to mechanistically think about how it ends up being stuck in the nuclear genome, what happens, or what's thought to happen, is that it's a byproduct of the DNA repair machinery. So, you know, if you think about molecular biology, you know, chromosomes are not perfect. Mutations are happening, chromosomes are breaking, and cells of all kinds invest a lot of effort into maintaining their chromosomes. So there are constantly DNA repair enzymes that are going around repairing breaks in nuclear chromosomes. So the irony is that it's the process of trying to repair nuclear chromosomes that gives rise to EGTs. Because if the repair enzymes are in the process of, you know, fixing a break, and there happens to be a little piece of organelle or DNA that's floating around, it can get incorporated into that DNA. So they're trying to fix it, but in some ways they make it worse. And I'm not just making this up, there's experimental evidence for this, and if you look very clearly at genome sequences, you can see lots of examples of very, very recent transfers. So there's a lot of mechanisms that are known that underlie this process. So organelle lysis, I guess, is the first point, and then somehow the DNA, just by chance, makes its way into the nucleus, and again, just by chance, in the course of DNA repair, chromosomal repair, occasionally the foreign piece of DNA will be integrated. But again, it's probably going to be dead. It's, you know, the system doesn't know to move genes in gene-sized pieces. It's just taking broken pieces of organelle or DNA and inserting it. And just by chance, if it happens to contain a whole gene, then it might be able to be expressed and take on a function. But almost always, it's a dead end road. I hope that helps.
Yeah, I think so. I know that you are not a specialist in chlorophyll, but we have a question that was put again here. Maybe you could just confirm that it's not your area.
Does chlorophyll have enough inner variability to not be used as a proxy for primary productivity? How can we use such a variable component for these estimates? Well, I guess by inner variability, you might be referring to, you know, its variations on a theme, the structure of the chlorophyll molecules, and that it might not, its presence might not in and of itself be used to indicate that a lineage is photosynthetic. That's how I might interpret that. And I don't, that might be true, it might not be true. The second part of your question, I guess I don't really understand quite. Could you put it up again, Melton? How can we use such a variable component for these estimates? I don't know that we know enough about the first part to be able to answer the second part. But nothing that I know about algal biology and biochemistry, and again, I'm only specialist in certain areas, nothing I know tells me that there are chlorophyll-like molecules in organisms that are using it for something totally different than photosynthesis. So I think there is a pretty good correlation between the presence of chlorophyll in its various forms, all of those different subforms we talked about earlier, and phototrophy. But again, I think a pigment, you know, a biochemist, a plastid biochemist would probably have a much better answer than I would. It's a good question, though.
At least I have a bold question for you. Actually, what do you expect? What are your expectations for the next 10 years about this kind of studies? What kind of discovery will we make? Something like that. And how could the Latin American, for example, to help in these discoveries? What do you think? Your opinion?
Okay, that's a really good question, and I appreciate it. Next 10 years, I do, I've been asked that before, and I sort of think about it in the context of grants and so forth. What I would say, you know, when I put up that slide and I said that, you know, eukaryotic genomics is in the same place that bacterial genomics was 20 years ago. To me, that's, at least people like me and people who are interested in the same sorts of questions, that's where the field is going to go. And what do I mean by that? If you think of the whole tree of eukaryotic life, what have we been doing primarily when it comes to sampling genome diversity space? Well, what did we do? We picked a diatom over here, and then we picked a haptophyte, then we picked a cryptophyte, and then we picked this, right? And that's the first step. If you want to better understand diversity and you want to use genomics as that lens into symbiosis, you pick all the lineages and you choose a representative. But one thing that bacterial comparative genomics has told us is that one representative just doesn't cut it. You know, there's thousands of E. coli genomes sequenced now, and they are all incredibly different. And so what we've learned is that when you sample genomic diversity at the tips of the tree, you learn some pretty amazing things. And so in my group and other groups, where we are going now is to use the latest sequencing technologies to do fine scale sampling. So pick an algal lineage and don't do one genome, do 10 of them, do 20 of them within fine space, you know, different strains, different species, different genera, and it gives you a picture of the diversity at the microscale, because that can help you understand the macroscale patterns. Because when you've only got one haptophyte and you've got a diatom and you've got this, when you're trying to distinguish between EGTs and LGTs, oftentimes you just can't do it. The data is not rich enough. So we are investing heavily in lineage-specific deep sequencing to get a picture, the fine scale patterns of genes coming and going. And then we hope to ultimately be able to present it to, you know, contextualize it in the bigger tree. Now, the second part of your question had to do with Latin America and what researchers in Latin America can do. I think there's lots of, that's just one avenue and one approach that I'm interested in, but I think the same kind of logic applies to other types of data. You know, it could be ultrastructure, it could be pigmentation and so forth. So I think there's a lot to be learned from specialists looking at individual lineages and learning about what's going on at that fine depth and then expanding it up. You know, for anyone in Latin America who's interested in genomics, if you are applying it to your own questions in your own algal groups, even if you're not a specialist, but you have lineages that you think are interesting from the perspective of symbiosis or plastid biology, I would encourage you to reach out to international partners, because there are a lot of initiatives that are happening. I'm part of a large initiative that's just getting off the ground with a collaboration between the Gordon and Betty Moore Foundation and the Wellcome Sanger Center in the UK. There's going to be hundreds of new genomes from symbioses of all different types, corals, animal microbe symbioses, all in the aquatic realm. And my part of it will be the microbial symbioses. But that's one way that people can participate, and those are open projects. So really, I'd encourage anyone to reach out and try and partner with other perspectives, because everyone's got their strengths and things that they can bring to a larger project. And it doesn't really matter necessarily whether you've got genomic expertise. The important thing is that the lineages get sampled, and the genomics and omics tools can be leveraged to help feed back on the organisms to develop genetic tools and all kinds of things. So that's the best advice that I can give.
Oh, good. Well, now we have just thanks for you, a lot of thanks of the participants here, and I also thank you very much for participating with us and to bring us this excellent presentation, excellent lecture.
Okay, well, it's been my pleasure, and I wish you all the best, and I hope you stay healthy and happy at what's a challenging time for everybody. But take care, everyone.
Then, bye, people. Até mais. Até a próxima apresentação, às 14 horas, com Clorota, com a professora Ina de Souza Nogueira. Bye.